And welcome everyone to the Global Immunotalk. My name is Matteo Iannacone and I'm one of the co-organizers of the Global Immunotalks. And I'm really excited today to host Lydia Lynch as our speaker. But before I introduce her, let me remind you to join next week when Steve Jameson is going to be our speaker. So it is a real pleasure to introduce Lydia Lynch, who I will ask to turn on her video. Lydia is actually an associate professor. Hello, Lydia, at Harvard Medical School and the director of metabolic core at the Brigham and Women's Hospital. Lydia is originally from Dublin, Ireland, where she did her PhD at University College Dublin before moving to Boston for postdoctoral training with Uli von Andren and Michael Brenner at the same time. She then got a junior faculty position there and she rose through the ranks. And then, as I said, she's now an associate professor. Lydia's research focus is to understand the interdependence of the immune and the metabolic systems, particularly in the setting of obesity, of metabolic disorder, as well as cancer. And she studies this with a variety of approaches, including intravital imaging, whole body metabolic analysis in vivo, multi-omic approaches of metabolic disorders in both humans as well as in mice. Her lab has provided key insights into the effects of obesity on the immune system and how this leads to reduced immune surveillance and reduced tumor control. For instance, she discovered that NK cells were depleted in human obesity, that NK cell dysfunction in obesity was due to a metabolic reprogramming towards lipid metabolism, which inhibits their ability to kill tumor cells. So her goal is to identify metabolic markers that can predict worse metabolic health and increased risk of cancer, and to develop methods to target immunometabolic pathways for protection against a variety of obesity-induced diseases, including infection, metabolic disorders, and cancer. So for her work, she has received numerous awards, but I would like in particularly to mention that she received in 2020 the Mentor Award from the Brigham and Women's Hospital Department of Medicine. So thanks so much, Lydia, for accepting to give a talk today. And before I leave the floor to you, it's a tradition at Global Immunotalks to ask a personal question to get to know our speaker a little better. And so what we would like to ask is, what is the best piece of advice you have gotten in your career?
Okay, thank you so much, Matteo and Elena, for the invite and for the introduction. I'll answer the question first and then I'll say thanks again. So I have got loads of great advice through the years of my career. The ones that's probably had the biggest impact on my career, though, is probably two. So it's not that they were really like wise words or anything. It was more of an attitude, and they've really stuck with me. So first, when I was finishing my PhD in Ireland, my PhD supervisor, my mentor, Professor Cliona O'Farrelly, she said, I had done well, she seen potential in me, and now I needed to go to America for a postdoc to get the best training that I could get. And I remember, thinking that she was mad. I had two children at this stage. I was looking after my dad. He lived with me. He was old. We had no money. I came from a different background and I really couldn't see myself in this scenario, but she could. And she said, I said to her, like, how am I going to go to America? Don't be so ridiculous. And she said to me, Lydia, you just will. I know if it was the way she said it or her belief in me or something, but I thought, right, I will. There was a shift in my head kind of, and I was able to make it work. So then I applied for the Marie Curie and the UNESCO, and off we went, the six of us, on one postdoc salary to live in Boston. And, it was the best adventure of my life and, maybe of my dad's life too. He was nearly 80 at the time. And so I know I wouldn't have done any of that if she hadn't said that to me. So it was great advice at the time. And then the second one was from my postdoc mentor when I got to America, Michael Brenner. And, the postdoc had gone very well and I applied for my K-99 to transition to independence and I didn't get it. And I was kind of dreading telling Michael. And anyway, I told him, and he said, well, Lydia, your career will be defined by how you deal with this when you have these failures, not by how what you do when the going is good. So why don't you feel sorry for yourself today, go home, commiserate, and make sure you come back tomorrow and you try again. And so I did, and I kept this advice in my head, every time we have one of the many failures that come with this job. So I think they were the two that were most important to me.
Well, thank you very much, Lydia, for sharing these inspiring stories. I'm sure they will help a lot of young trainees facing adversities. So without any further ado, you can now share your screen, and I'm really excited to hear your talk entitled Understanding the Paradoxical Effects of Obesity on the Immune System.
Okay.
Lydia.
So you can see all okay now, Matteo?
Yeah, works well.
Okay, great. So thanks again to Matteo and Elena and all the organizers, for doing this for the immunology community. I don't think I've missed the talk yet. If I've missed them when they were live, I caught up with them later on YouTube, and I really think it's brought us together and also reminded us to be open the way science should be. And then second part is thanks for inviting me. It's a huge honor for me, and so thanks again. And as Matteo said, I'm going to talk about our work through the years on the effects that obesity has on the immune system. So I'm just going to keep doing that. So for this type of talk, I thought I'd start with how I became interested in obesity as an immunologist. And I suppose it was really because of this person here, Professor Donal O'Shea in Ireland. So Professor Donal O'Shea was moving back from the UK to Ireland to set up Ireland's first hospital-based weight management clinic. And, he had to build everything up from scratch. We didn't have any obesity services at the time. And so I had started hearing about it and, reading about it and reading how obesity wasn't just associated with the things here in blue, like diabetes and insulin resistance and fatty liver disease, but also how it was associated with all these immune-associated complications and diseases, like, increased risk of infection, poorer vaccination outcome, increased arthritis, and then an increased risk of many different cancers. And so as a new immunologist, I thought that this was really fascinating and, something must be wrong with the immune system. And so Donal also wanted to start a lab in Ireland, and he advertised for a Newman Scholar, which I applied for, and I got, and he basically let me pick what the topic was that we would study in the lab. And so I picked, what's gone on with the immune system in obesity. And although I wasn't there in his lab for that long, because I got the fellowship to go to America, I've been really very lucky to continue working with Donal for the whole time that I've been away. And so we're trying to answer this question together. So what do we know about the immune system in obesity? At the time, it was emerging that adipose tissue had its own immune system, and that this was very changed, dramatically changed in obesity, leading to increased inflammation. So you had all these regulatory cells in normal lean adipose tissue, and as you gained weight, you got an expansion of macrophages and a change in the lymphocyte compartment and other things. And this led to increased TNF-alpha and inflammation locally, but also systemically. And this work really was started by Gökhan Hatta-Mishlegel in the Science paper, where they showed that increased TNF-alpha was up in obese adipose tissue, and that it actually interfered with insulin signaling and so contributed to the metabolic disease. And so together with Donal and Michael Brenner and others, we found, we were studying this as well, and we were looking at protective cells like iNKT cells in adipose tissue that are lost in obesity. But I'm not going to talk about adipose tissue immunology today. I just want to mention it so that to mention the low-grade inflammation that's going on in the background. So what else did we know about the immune system in obesity? We knew that people with obesity have increased risk of infections. This was very obvious during the H1N1 in 2009, and again now in 2020 from our current COVID pandemic. We know people with obesity are more susceptible to severe symptoms and also increased death. And this is clearer than, clearer in young people than in old people, because old old age is obviously a factor for severity and death in COVID. But when you look in the young people, you see people with obesity have a much greater risk of severity and mortality. And it wasn't just respiratory infections. We know that there's an increased risk of post-operative and other hospital infections with obesity, a much greater risk of skin infections, pneumonia, uterine tract infections. There's people with obesity often require longer treatment for infections. They get more serious complications. And then if you're obese with diabetes, this can double the infection rate again. Now, we also know, similar is that obesity is linked with cancer. There's about 13 cancers that you have an increased risk of if you're obese. And top among these are endometrial cancers. Half of endometrial cancers are due to obesity, but also all of these cancers and more. About 500,000 cancer cases worldwide a year are due to obesity. And despite this, actually, a lot of the general public don't know that there's an increased risk of cancer with obesity. Most people know that smoking causes cancer, but most people don't know that obesity causes cancer. And so, scientists like myself and others, we probably need to do a better job of getting that message out to the general public. So this increased risk of infection and cancer suggests that there's a compromised immune system in obesity in humans. And so this might suggest maybe obese people have less autoimmunity. This is not the case either. There's a big increase in the risk of autoimmunity with obesity as well. So if you look here at rheumatoid arthritis, for example, the odds ratio is 1.2 to 3.4. So there's 1.5 to 3 times greater chance of getting rheumatoid arthritis if you're obese. MS now has an odds ratio of 2. So obese people are twice as likely to get MS. And I think the highest one, or one of the highest ones, is psoriasis and psoriatic arthritis, where people with obesity are 6 times more likely to get psoriasis and psoriatic arthritis. And then with all of these diseases, there's also an increased severity and increased symptoms that happen with these infections and autoimmunity with obesity. So you kind of on first thought, it seems like a paradox. You have an increased risk of cancer, increased risk of infection, suggesting that the immune system is suppressed. Yet you also have an increased risk of autoimmunity and also a chronic state of low-grade inflammation going on. So what can unite these things? And we don't know the answer to this yet. Well, possibly metabolism, and possibly mitochondrial metabolism, and how changes in the whole body metabolism influences the immune system metabolism. And so that's what my talk is going to be about today. Our lab works on linking systemic metabolism with cellular metabolism, and the field of immunometabolism is kind of split into 2 sides, and those that work more on cellular immunometabolism, which is studying the different bioenergetic pathways that the immune cells use to fuel their different functions. And then on this side, systemic metabolism, systemic immunometabolism, kind of the unexpected role that the immune system plays in regulating the whole body metabolism, but also how when the whole body metabolism is changed, like in people with cancer and in people with obesity, what effects this has on the immune system. And what we're particularly interested in is linking the two, because they're both dependent on each other. When you've got systemic metabolic changes that are going on, this alters the metabolites, the fuels, nutrients that are available to the immune system to use as fuel and as building blocks for their function. It changes their function. And when their function is changed, that has an impact on whole body metabolism. And so I'm going to talk about this kind of interdependence, and well, particularly today, just in the setting of cancer. So there's loads of different reasons why there's an increased risk of cancer in obesity. There's inflammation, there's increased lipids, there's insulin signaling, glucose, adipokines. All of these can directly impact the immune system, or sorry, the tumor cells. But what's left out of some of the reviews is what is the effect on immune surveillance. And so this is the first thing that we worked on together with Donal in Ireland about 10 years ago or more now. In humans, we started to look at NK cells. We started there because NK cells are our first line of defense against infection and cancer. And we found defects in NK cells in people with obesity compared to people who are lean. If you put NK cells, so with tumor cells, they kill from people who are lean versus obese. They kill less tumor cells and they produce less interferon gamma. And so then we went on more recently to say what Matteo had introduced, that we actually think it's the metabolism of the NK cell that's changed, probably caused from systemic metabolism changes, that is really affecting the NK cell killing. So I'll just talk a little bit about that. So to get more at what was happening to the NK cells, we sequenced NK cells from lean and obese people and also mice. And one of the things that we see is that there's this decrease in cytotoxic machinery. So there's a decrease in granzymes and perforins, the things that the NK cells need to kill. And there was an increase in all these lipid-related genes. So increase in CD36, you know, for fatty acid transport, fatty acid binding proteins, LDL receptor, lipases, CPT1, CPT1A and B. Interestingly, these are all PPAR target genes, the transcription factor that controls lipid metabolism. And so because of these changes, we started to look, well, have NK cells switched to a lipid metabolism or to take up lipids? So we looked at NK cells directly from people who are lean or obese, and using the LipidTox stain, neutral lipid stain LipidTox, we see that the NK cells stained greater for lipids. And when we looked by confocal, it wasn't the membrane, it was that there was lipid droplets inside of the NK cells, which NK cells don't usually have lipid droplets. And so this is kind of what I was trying to say in the introduction. We think, you know, if NK cells are what we eat and maybe immune cells are what we eat, and if you give them a lot of fatty acids, then they'll maybe take them up and try to use them and maybe metabolically reprogram themselves. So the question then is, well, can NK cells use the lipids as fuel or for their normal function? So first thing we did was take NK cells from lean people and give them palmitic acid and oleic acid. These are lipids that are increased in people with obesity, and they're also the main component of the high-fat diet, typically used for mice in the lab. And the first thing we see is when you give them NK cell, or when you give NK cells lipids, they take them up. And I suppose not too surprising now, there was a decrease in glycolysis of the NK cells, but it was a bit more surprising that there was also a decrease in OXPHOS. So, you know, it didn't seem like they were doing fatty acid oxidation. And so it kind of metabolically paralyzed them. And then we went on to take NK cells directly from people who are lean or obese. This is work we did in Dublin with David Finlay and Andrew Hogan. And when you take NK cells, they're quite, you know, quiescent, they're not doing much. When you activate them, they ramp up both glycolysis and OXPHOS. But the NK cells that were from people who were obese, they couldn't turn on glycolysis or OXPHOS. They were essentially metabolically paralyzed. When we measured their ATP levels, they really didn't have any energy. But they weren't dead. They were still alive. They just had no, you know, spare metabolic pathways. And so this was something that the question then became, well, how does this impact them? What do they need their metabolic pathways for? And this was something of interest to myself and Xavier, who was my first postdoc. He was a cell biologist. And so he wanted, you know, he loved thinking about what was going on inside the cell. And we learned a lot from Jordan Orange about you know, how the NK cell sees and kills the tumor. And so it's a very stepwise pathway. NK cells have this microtubule organizing center and all of their lytic granules in the cell. And when they see the target cell, they form a synapse, they rearrange their actin filaments, sends an activatory signal into the cell. All of the lytic granules come together to the mTOC, and then the mTOC brings everything to the synapse where the NK cell kills. And this is really important for the tumor cell to be killed. It can't just, you know, NK cells can't just release their granules everywhere. It doesn't work. So we can image this by using pericentrin to measure the, to look at perforin to measure lytic granules. Pericentrin is a measure of the mTOC, and CD2 measures the, visualize the synapse. And so when we do this, we see that when you put an NK cell, that's the little fella here with the tumor cell, it has formed a synapse, and the pericentrin is at the synapse and the tumor cell dies. But when the NK cell is first treated with lipids like palmitate, the NK cell is able to see the tumor, it's able to go over, it's able to form a synapse, but the pericentrin is at the back of the cell, and it doesn't polarize its lipid granules to the tumor, and so it doesn't die. So then we did this directly ex vivo with people, NK cells taken from people who are lean or obese. Here's the NK cell with the tumor, it's formed a synapse, the pericentrin is at the synapse. But here is one with 3 NK cells attached on. There's the 3 of them have formed the synapse. In all cases, the pericentrin is at the back of the cell. And so we found that I'm not shown it here, but glycolysis and mTOR pathway was important for, not for the NK cell to move towards the tumor or to form the synapse, rearrange the actin filaments, but it was important to polarize the lipid granules to the tumor. And without that, then it can't detach and go on and kill loads of other tumors like the serial killer that it is. So we think that this is a major factor for why NK cells don't work as efficiently in people with obesity. So the question then is, does it mean more tumor growth? And we couldn't really measure that. We couldn't really ask this question in mice. So in vitro, we see, yes, NK cells, when they're treated with lipids and they're metabolically paralyzed, they don't kill tumor cells very well. But we needed to go to a mouse model to see if it was true. And so this is when Lydia Lynch joined my lab, and she looked at in vivo at the cytotoxicity. So here she did 2 tumor models, B16 and MC38, and both of them have increased tumor growth in high-fat diet. This is Lydia when she started and during it, and then we submitted the paper, and then during lockdown she had twin babies and managed to get the paper published and get a Marie Curie. So she's now a Marie Curie Fellow in Berlin, and I'm sure she's soon to be a junior or young PI, because she really is fantastic. So then she looked in the tumor to see what was happening with the NK cells, and surprisingly, we wasn't just NK cells that were affected by obesity. There was also less CD45s in total, less CD8s, less CD4s, and less NK cells in the tumor. And when we looked at their phenotype, of course, CD8s and NK cells are already a bit inhibited in the tumor because of the suppressive microenvironment, but this was further reduced in obese tumors. So there was less interferon gamma. It was already low, and it was really even much lower here, less granzyme B, less proliferation, less phosphorylation of S6. And so it seemed like there was a kind of a cold tumor phenotype in obesity. So because of this cold tumor phenotype that we see in both B16 and MC38, we wondered if we could see if this was also the case in humans. And so we collaborate with another Donald, Professor Donald Brennan in UCD in Ireland, and we looked at endometrial cancer because, as I mentioned, endometrial cancer is one of the top ones at risk in obesity. And so we looked at histology here, and here's one patient whose BMI is 26, another patient whose BMI is 46, and they both have the same type, same stage and grade endometrial cancer. And you can see there is T cells in both patients infiltrated into the tumor, but there is more CD8s and more PD-L1 in the endometrial cancer from people who are lean. And so if we just look, that's just one person, if we look at more people, we see that there seems to be a reduced infiltration of CD8 T cells when you have an increasing BMI in endometrial cancer. So we have a little bit more work on this, which I won't have time to go into with weight loss, but you know, at least from this first look, it looks like there could be a colder tumor phenotype with obesity in humans as well. So the question then is all immune cells, you know, defective in obesity? And this doesn't seem to be the case. It seems that Tregs, for example, love obesity and love, at least in the tumor, love lipids. And so this was work done by Ping-Chi Ho. And then more recently, this really elegant studies by Hongbo Chi. And what Ping Chi found was that Tregs in human breast cancer were enriched for fatty acid metabolism compared to Tregs in matched blood. And they had also upregulated CD36 compared to in the blood, and they had taken up more lipids. And when you knock out CD36, specifically on the Tregs, they didn't accumulate as much in the tumor, and you had some better tumor control. And then what Hongbo found was that Tregs can also, in the tumor, make their own lipids, and this was very important for their survival and their suppressive function. So it's not all immune cells that are the same. Sorry. And so then we also had been looking at IL-17 producing cells, and they also seem to like lipids. And so this was a really collaborative project with Professor Bruno Silva Santos from Lisbon, who recently gave the Global Immunotalk, and also Dan Pennington from London. It was actually an accidental collaboration when we all realized we were working on the similar kind of things. Rather than compete with each other, we decided to bring the work together. And so using gamma delta as an example of a distinct IL-17 cell to study, in my lab, Claire and Mathilde did this work mainly. And so gamma deltas in mice are very distinct, either IL-17 producers or interferon gamma producers. And the ones, and you know, loosely IL-7 gamma deltas that make IL-17 have a pro-tumor function, whereas the ones that make interferon gamma have an anti-tumor function, like NK cells and CD8s. And when we looked at these cells, these two types of gamma deltas, we see loads of lipid droplets inside ones that make IL-17, but not inside ones that make interferon gamma. And it wasn't just gamma deltas, it was other cells that make IL-17 as well. So we see that the ones that make IL-17 take up more lipids, they have more mitochondria, they have more mitochondrial potential, and just evaluating the amount of lipid droplets, like they have way more lipid droplets inside of them. And so Claire analyzed what was in the lipid droplets, and she found it was triglycerides, but also cholesterol. And they have special machinery to take these lipids up, when we found from sequencing them. And so we wonder, well, we know that there's more fatty acids and cholesterol in people and mice with obesity. So what would happen to the gamma delta 17s in obesity? And we found that there was a huge expansion of gamma delta 17s, whereas there wasn't in mice with obesity, whereas there wasn't in the interferon gamma producing ones. And also in Yale with Vishwajit Dixit and Emily Goldberg and Akiko Iwasaki, they also found an expansion of gamma deltas in ketogenic diet. So it seems IL-17 producing cells like lipids too. And in the tumor, there's a more lipid-rich environment. And we see the ones that make gamma deltas that make IL-17, they're usually the minority. Most of them make interferon gamma. But in the tumor, there's an enrichment of the ones that make IL-17. Potentially, lipids are playing a role here. And so then we did the tumor model in obese mice. So we fed them a standard fat or a high fat diet, injected B16. Again, the tumor grows faster in obesity, and we see a further increase in IL-17 gamma delta accumulation in obese tumors compared to lean. So then we wondered, well, can we manipulate lipid metabolism and to get a decrease in the gamma delta 17s, because they're not something you want to have in the tumor. So we did this with orlistat, which is a lipase inhibitor, to block the uptake of lipids. And when we see this, we see a decrease in the accumulation of gamma delta 17s in the tumor. We see that they had less lipids inside of them, and that the tumor growth was suppressed. Now, obviously, this was into the whole body, and so tumors also like lipids, and so we couldn't rule out that it was the tumor that was not able to take up the lipids. So then what we did in collaboration with Bruno's lab and Noel Lopez, we expanded the gamma delta 17s in the presence of cholesterol before the adoptive transfer. And you see when you inject in gamma delta 17s, they increase tumor growth a little. But when you pre-treat with cholesterol, they had much more IL-17 production, and the tumor growth was really enhanced. So, it seems Tregs and IL-17 producing cells love lipids, and these are both cell types that you don't want to have in the tumor. So just as an aside here, just a quick aside, we're wondering here now, this IL-17 finding enhanced in obesity, could this relate as well to both the increased risk of cancer and autoimmunity in obesity? So, you know, we see increased IL-17, lots of cells that make IL-17 with obesity. This other group here, Nakayama's group, found also Th17 cells are expanded in obesity. And Andrew Hogan in Ireland, who's a good friend of mine now as well, made cells also polarized towards IL-17 in humans. So there is a lot of accumulating evidence that there's increased IL-17, so it could relate to the autoimmunity, and whether this lipid finding could be something that is similar to both the pro-tumor and autoimmunity needs to be worked out. So back to the tumor. And what effect does obesity have directly on the tumor? So we found that, you know, it's suppressive to the immune system, but what is it doing to the tumor itself? When we eat food is converted into blood sugar, rising glucose in the blood signals to the pancreas to release insulin. Eventually, insulin resistance can develop if this keeps happening. And then you have glucose buildup, and it can lead to weight gain. In obesity, there's increased free fatty acids and TNF alpha, as I mentioned earlier. And then there's other factors that cause insulin resistance. And then you get this compensatory hyperinsulinemia. And this increase in insulin levels then upregulates hepatic IGF-1 production. And then the downstream of both insulin signaling and IGF-1 signaling is PI3 kinase, AKT. And, you know, so insulin and IGF-1 signaling promote cellular proliferation. They inhibit apoptosis in many tumor types. And so this work is really, a lot of the work is done by Luke Hantley. And, you know, it's clearly something that's happening to tumor cells in obesity because of the glucose and insulin changes. But so there's a lot of research into trying to reduce glucose and insulin so as to reduce tumor growth. But then this paper here by Matt van der Heijden in MIT in Nature last year, they showed that they looked at both caloric restriction and ketogenic diet. And they showed that in both diets, you have a reduced blood glucose and reduced plasma in both, but only the caloric restriction condition reduced tumor growth. Whereas the ketogenic diet didn't. So this adds another level of complexity, that it's not just, the tumor growth is not reduced only by reducing glucose and insulin. And why is this? So this is because, lipids accumulate in the tumor, in the interstitial fluids, in the mice that were fed ketogenic diet. So this here is showing the different fatty acid species compared to standard fat diet. And you can see that the caloric restriction, you've actually got a reduced amount of lipids in the tumor, whereas in the ketogenic diet, despite having a lot less glucose and less insulin, you've got more lipids, and the tumor's low lipids. And so when they removed the lipids from the media, for example, then the tumor cells grow slower, but actually what they do is turn on lipid synthesis. So if you stop lipid synthesis and lipid accumulation, then the tumor cells slow down their growth. And so therefore, you know, because tumors like lipids, this was work done by my friend Samir Bayaz, Nature paper while he was a PhD student in Harvard. He found then by feeding mice a high-fat diet, it increases the stemness of tumor cells. And it was also because they turned on a PPAR-driven lipid metabolism pathway. And Salvador Benita in Barcelona also found this in human and mouse breast cancer. Cells that metastasize away from the original site to the secondary site, they have CD36, another PPAR target gene, and they use lipid metabolism. And this was important for their survival. So if we could put all of this information together, could lipids be the common feature of a pro-tumor immune response? So we know in obesity, you've got increased free fatty acids. It can come from the diet, it can come from adipocytes bursting, And the lipids go everywhere. They go to the liver, the pancreas, vessels, muscle, and they can cause insulin resistance. But now we know also tumor cells can take up lipids in a high-fat diet, and so can the immune system. But in the case of the tumors seem to love them. They enhance their proliferation and metastasis, whereas in the case of the immune system, NK cells, CD8 T cells, do not like excess lipids, and it impairs their function, whereas Tregs and gamma delta 17, so other IL-17 producing cells that have a pro-tumor function, are enhanced in obesity. And so we wonder if targeting immunometabolic pathways might be beneficial to improve immunity in metabolic disorder. And this is something that we're working on, including PPAR targets. So one of the questions that we had as well as we went along was, will immunotherapy work in patients with obesity? And so we know from the literature that PD-1 signaling can block glycolysis, it can enhance fatty acid oxidation, obviously block TCR signaling. And given that blocking PD-1, PD-L1 can restore glycolysis, we hypothesized that this wouldn't work in people or mice with obesity because they already start off with a metabolic dysfunction. And so we thought that, anti-PD-1 therapy will not be strong enough to reverse this metabolic defect. So again, Lydia did this work, and we first looked in the B16 model. We fed them a standard fat or high fat diet. B16 is not controlled on its own by anti-PD-1, so we did it with a PD-1 vax and a vax. And we see that PD-1, the treatment PVAX, works very well in the standard fat diet, but it also worked equally well in the mice that were fed a high fat diet. So far, immunotherapy did work pretty fine. And when we looked at the immune cells, we see that there was, very few infiltrating CD8 T cells in obesity. This was enhanced in the standard fat diet after PVAX, but it was even further enhanced in the mice that were obese. And it was able to restore interferon gamma, or increase a little bit, but it was really able to restore interferon gamma production by CD8s in the obese condition. So we were very surprised about this. We were very happy about this because of the potential implication for people with obesity. So we looked in another model, MC38, because this one is controlled by PD-1 alone, and we did the same thing, fed them standard fat or high fat, and then give the anti-PD-1 treatment. And we've seen that no matter whether the mice were lean or obese, the checkpoint therapy completely reversed or completely got rid of the tumor growth. And so because we had no tumors to measure here, because it was completely gone, Lydia left the mice for 4 months and then challenged them again. And so on the second challenge, which is a memory challenge, both the standard fat diet fed and high fat diet fed were able to completely block the establishment of the tumor. So it seems like the checkpoint therapy is really powerful to overcome the metabolic defects seen in obesity, and it doesn't affect, or at least the checkpoint is able to reverse the T cell memory to the MC38 tumor. So we were hoping then, after getting these results, to see from the literature, from patient charts, if this was the case in humans as well. But while we were doing this really cool paper came out by Jennifer McQuaid in the Lancet, and she found that, and her team found that obesity actually was not detrimental for immunotherapy. If anything, it was good for immunotherapy. So here was melanoma that metastasized to the lung, checkpoint therapy, and here the normal BMI, people with a normal BMI had a worse overall survival than people who were overweight or obese when they were given immunotherapy, And this is not the case with chemotherapy. So what it looks like is that immunotherapy is not, or obesity is not detrimental for immunotherapy. And if anything, although we can't say we can't say this worked better in obese mice because it worked equally as good in both. But the delta here to go from black to blue versus red to orange is much greater. So it really, this is just to bring up another paradox. And so although you get increased risk of cancer, potentially immunotherapy works even better in people who are obese. And so this is really becoming a very interesting question in the field that there's currently no really good answer for. So, you know, for young people out there, this is an exciting thing to study, I think. Okay. So if we go back to this is, you know, coming to the end, lipids as a common feature of a pro-tumor immune response. So then we started to wonder, well, you know, is it all lipids? And so Hannah, a PhD student, took this project on in the lab, and it's really become bigger and bigger and bigger. So now we have a team of people working on different aspects of this project. But to see if all obesity is the same or all high-fat diet is the same, Hannah fed the mice a high-fat diet from which was all of the high-fat diets were isocaloric, and they all had the same amount of carbs, proteins, vitamins, and minerals, but the source of fat was from a different place. So either lard or butter, the animal sources, or palm oil, peanut oil, and coconut oil. We've also done olive oil as well. And the first thing we see is no matter what you feed a mouse, you know, what source the high-fat diet comes from, all the mice become obese. And it wasn't that they had a kind of, you know, weight put somewhere else. All the fat pads were equally much increased compared to the standard fat diet. Sorry, this keeps jumping. So then we went and did the tumor experiment again. And again, here we used B16. And lard is the typical high-fat diet that people use. So this is, again, when we feed the mouse lard, you have accelerated tumor growth. When we fed the mouse coconut oil, they didn't, though. And peanut oil, you know, it was slightly increased, but it wasn't as dramatic. But the two that were the most different to each other were butter and palm oil. So butter was always had the greatest tumor growth, and palm oil always had the lowest. Slightly under, it wasn't significant, slightly under the standard fat diet. So this is just one time we did the experiment, the tumor volume. And this is despite the mice being, all the mice there being equally obese. So the point of the study was not to learn from a mouse what fat we should eat, because they're only one source. It's not realistic, but to see, can we learn something from, you know, what's happening metabolically from the worst versus the best high-fat diet. So then we went on to pick butter and palm to compare, and we see they both eat mice fed butter versus palm. They gain equally the same amount of weight. It's not an apple pear situation. They have equal amount of gonadal distribution versus subcutaneous distribution of their adipose tissue, and all of them is much bigger than the standard fat diet. But the tumor grows faster in the butter than it does the palm oil. So we look in the tumor, we see that there's a poor infiltration of NK cells and CD8s in the butter fed, but not the palm. It was lower, but not significant. And there was less interferon gamma production by NK cells and CD8 T cells in the butter, but not the palm. We see again that, and this was really consistent, that as I showed before in humans, we also see in mice, in the circulation, immune cells take up lipids or become accumulated in them. And so we only seen that, though, in the NK cells from mice fed butter, but not palm. This was NK cells. We also see the same for CD8 T cells and for gamma delta T cells. They have increased lipid tox only from the butter, not the palm, despite equal obesity. And this was really striking. This, I think, was done by Roshin in the lab, when we isolate the NK cells from mice. This is in a non-tumor setting, from mice fed butter or palm. You see the same metabolic paralysis we've seen before, no glycolysis, no oxphos being able to turn on. However, the mice fed palm oil had totally normal metabolism. The NK cells had totally normal metabolism. From mice fed palm oil, even they were super obese compared to the standard fat diet, both for glycolysis and oxphos. So we thought, well, there's obviously something big going on that we don't know about, so we'll put them into the metabolic cages, and this, you know, maybe the mice are not behaving as if they're obese at all. This wasn't the case. There was no gross differences. When mice are put into metabolic cages, you can measure their respiratory exchange rate. This is a measure of the fuel that they use as the fuel source, the source of fuel they use. So, you know, a mark of 1 is they're burning mainly carbs, 7 is they're burning lipids. And so this is circadian. When they are awake eating chow, they're burning carbs, then they go to sleep, they burn lipids. And when the mice are obese, they switch to lipid burning all the day long. And this is a feature of obesity. And we didn't see any difference in this between mice fed butter or PAM. And we also were surprised to see, we thought then maybe this is an example of metabolically healthy obese, but it seems that they also had impaired glucose handling, whether they were fed butter or PAM compared to standard fat diet. So we then wondered, well, is there more subtle changes in the systemic metabolome going on that we can't see in the metabolic cages? And so we collaborated with Ivana Mills and Ed Chucheni in the Dana-Farber. Ivana has just started her own lab in the Dana-Farber, and we did metabolomics on the serum. And so again, the first thing we see is so many things are similar between butter and PAM. Two obvious ones, aminobenzenoic acid and hippurate, completely decreased in the serum of mice fed any high fat diet. It's probably a measure of reduced microbiome diversity. But we were able to see some differences. And so mainly the difference here I'm shown is increase in PAM compared to butter, but there was really not much differences in these things here compared to standard fat diet. So it was the PAM didn't have any defects in glycolytic intermediates or TCA cycle intermediates, whereas the butter fed mice did. And the other thing that we seen was that there was an accumulation of oxydecanol carnitine and myristoyl carnitine. So these acyl carnitines were appearing in the serum, in the mice fed butter. And so we really were trying to say, okay, so what is the big difference between a butter fed diet or a butter diet and a palm oil diet? And when we look at the differences in all the diets that we used, actually butter and PAM are probably the most similar to each other in terms of saturated and monounsaturated and polyunsaturated, because some of the others were really different to each other, but these had the biggest outcomes. And when we looked at these different things, we couldn't really pinpoint the results to one type of fat. So we couldn't say for sure, oh, it's because there's no saturated fat in palm oil. Obviously, there's loads of saturated fat in palm oil as well, like butter, although there is more monounsaturated. So this ratio may be important. So then we looked at the fatty acid composition differences. Again, we see so much palmitic acid, which is typically thought to be bad for the immune system, that's in palm oil, and lots of oleic acid. And there was also lots of both of them in butter. But we did notice there was more stearic acid in butter and more myristic acid in butter. And so when we think back to the things that were increased in the metabolomics, oxydecanol carnitine is the acyl carnitine from stearic acid. Myristoyl carnitine is the acyl carnitine from myristic acid. So this kind of tells us, what we put into our mouth or what the mouse puts into his mouth can show up in the serum metabolome. And so then we went and looked at lard, because lard also has a decent amount of stearic acid. And so when we did the metabolomics, we see, although it also has a lot of palmitic acid, you don't see C16 acyl carnitine of palmitic acid in the plasma. You do see octadecanol carnitine. So you've got this increase in long chain or stearic acid or octadecanol carnitine in both butter and lard, but not in palm and also not in olive oil. So this is something then that Hannah started to think about, and she worked out that this may be incomplete fatty acid oxidation. So, because of the lipid overload in obesity, fatty acids going for oxidation in the mitochondria, well, they have their distinct fatty acid species with an acyl CoA. But the CoA can't cross the mitochondrial membrane, so it has to be carried across with carnitine acyl transferase, CPT1, in the mitochondrial membrane, exchanges out the CoA for the carnitine, and then you have an acyl carnitine. And then the next step is the acyl carnitine goes into the inner membrane by CACT for then CPT2 and fatty acid oxidation. And in obesity, you often see an increase in CPT1, and that's probably because there's so much lipids, bombarding the mitochondria. But sometimes you don't see an increase in CACT, and then you don't get the completion of the fatty acid oxidation. Instead, you've got an accumulation of acyl carnitine stage, and if they can't be oxidized, then they get pushed out of the cell as acyl carnitines. And so this is a measure of incomplete beta oxidation. And this has been shown in this really important paper that to happen in obesity. And, you can also measure acylcarnitines in the serum as a measure of, in the clinic, as a measure of inborn errors of fatty acid metabolism. So what we think is that these accumulation of long-chain acylcarnitines suggests that there's incomplete fatty acid oxidation going on, perhaps particularly from if there's a lot of stearic acid in the diet, which takes longest to oxidize. We have further evidence that this is what's happening. Again, you see a little increase in CPT. So the whole body incomplete fatty acid oxidation, we think, is probably coming from the liver and the adipose tissue. So we looked at both liver and adipose tissue, and we see this is, I think, just liver, but we see the same for both. There's an slight increase in CPT1A and obesity in both the mice fed butter and PAM, but for some reason, still unknown, there's only an increase in CACT in the mice fed PAM, not butter. And this, then you have this ratio where there's too much CPT or not enough CACT for the CPT1, suggesting, again, incomplete fatty acid oxidation. So lastly, we wonder, well, what does this, now you've got the accumulation of acylcarnitines in the serum. The immune system is not usually exposed to high levels of this. What impact does it have on the immune system? And so we cultured the CD8 T cells with different amounts of octadecanol carnitine, and we see it didn't impair their viability at all, but it really impaired their ability to produce interferon gamma. And when we look at all the different chain length acylcarnitines, we see the longer the chain, the worse it is. This is like almost too perfect. So C8 didn't do anything, C14 impaired interferon gamma, but worse was palmitate, and further was the steric derivative octadecanol carnitine. We also then looked at, well, what was happening to their mitochondria or to their metabolism. And you often see this depolarizing mitochondria in the tumor, as Ping Chi Ho has shown as well, and others. And so when we cultured the cells with, and given that their viability is fine, you start to see a decrease in the mitochondrial potential and accumulation down here. And this was even more striking with octadecanol carnitine. And so this suggests, well, there might be mitochondrial dysfunction. Is there ROS production? We didn't see much ROS production with C8 or C16, even though it impaired interferon gamma, but ROS did accumulate when, or was produced when CD8 T cells were cultured with octadecanol carnitine. Further, we've seen that the longer the chain, the worse impact it had on metabolism. It looks like shorter chains are fine. If anything, is a little bit boosted, but the palmitate version and the stearic acid version really impairs both glycolysis and oxphos. So, our hypothesis, which is still a working hypothesis, is that there's something about animal fat that causes incomplete fatty acid oxidation, kind of whole body. And, perhaps it's stearic acid in combination with something else. But when you have a lot of the longest chain fatty acid going into the mitochondria, and then you've got the CACT is not being upregulated, you've got an accumulation of octadecanol carnitine, which now is in the tissue, it's in the serum, and is exposed, the immune cells are exposed to it. It causes mitochondrial dysfunction and their reduction in interferon gamma. Perhaps why the animal is worse than the PAM in tumor control. And, this is obviously speculation. I have no evidence for this, but it does make me wonder, could there be an immunometabolic basis for going vegan at certain times of your life, such as maybe undergoing immunotherapy when you really need the immune system to work? Would vegan be beneficial? This is I'm sorry I haven't referenced here. I'm very sorry about that. This is the metabolome of people that either eat meat or fish or vegetarians or vegans. And you can see there's a lot of similarities here, except for vegans. Vegans have a really different metabolome. And this paper is a really nice paper, showed that acylcarnitines were highest in the meat eaters and lowest in the vegans. So, so is this something that could be done for people that are particularly at risk of cancer, such as people with obesity? It certainly seems easier to implement than suggesting weight loss, because weight loss is virtually impossible for so many people with obesity. Okay, so just to conclude, what does this all mean? It doesn't mean, you know, butter causes cancer, palm oil doesn't, definitely not saying that. But what we've done here is uncoupled obesity itself from the high-fat diet. And we see here when we make a diet with the exact same amount of fat, 45% of the calories coming from fat, the mice gain exactly the same amount of weight, they're all obese. You can still have profound differences in the metabolism in the cell and in the whole body in terms of metabolism. So with that, I just like to say thank you. Thank you to everybody in my lab, Gurv Mahagut, as we say in Ireland, to the people in the lab worked on this project and in the past and currently. You know, I've been really lucky to work with you, to our collaborators, Ivana and Ed, to everybody in Trinity UCD, especially Dave Finlay, Donal and Andrew, to Linda Sinclair in Dorian's lab in Dundee, Barb Nikolajczyk, Marie McDonald for samples, and my mentors, Clean O'Farrelly and Michael Brenner. And lastly, to the patients. There's a lot of stigma associated with obesity. Of course, there shouldn't be. The patients don't have to take part, but they do. And, you know, we wouldn't know any of this information without them. So thanks, everybody, for listening as well.
Thank you so much, Lydia. That was fantastic, fantastic. Yeah, I learned a lot and very, very exciting data on how fat coming from different diets can affect differently your immune system. So thank you so much for sharing your data and your very interesting perspective on the data. So appreciate it.
Thank you.
So as always, questions are via Twitter, and so if you have questions for Lydia, search for the account Global Immunotalks and then find the tweet that says ask questions for Dr. Lydia Lynch here, and then reply to that tweet with the hashtag Global Immuno, and Lydia will use her handle written here to answer those questions. So always remember the questions we do via Twitter because they allow us to do it in an asynchronous manner, and so people that watch these talks in YouTube can also ask questions. So, and thank you, Lydia, for accepting to answer the questions in Twitter. We appreciate that as well. Yeah, so with that, we will close the session. I have written in the chat the link to the tweet for the questions as well. And so, yeah, enjoy the interactions with Lydia in Twitter. So thank you, Lydia, again. And looking forward to next week, to the next Global Immunotalks by Steve Jamison. Thank you.
Thank you. Bye-bye.
Bye, everyone.